The epigenome is one word, but the epigenome is made out of multiple different chemical modifications in the DNA and the histones, the proteins that are able to pack the DNA, that help to pack the DNA. So the first study was DNA methylation. That was only one epigenetic mark. In this study of the reference epigenome of the disease, we actually measured in the same patient sample 9 different layers of epigenetic information. We developed computational tools to integrate all these layers, and through this integration what we actually generated was a functional map of the genome. So the genetic sequence itself is just information. Through our approach, we could segment or we could classify every region of the genome into a functional state. In total, we classify the genome into 12 different functional states of the disease. And then we said, well, now that we have these maps of the disease and of the normal B cells, let's compare them and let's see what changes in CLL as compared to the normal B cells. And we identified a total of 500 regions that are closed, are not expressed, are silent in the normal B cells, but in CLL they open. And through this opening, this leads to overexpression of several genes that were reported to be important for the disease, but were genes that were not associated to any genetic change, no genetic lesions, but now we identified there were functional alterations in the genome of CLL. And this 500 regions was a great finding, because this, in spite of the genetic heterogeneity, where we have like 5 mutations present in 5 to 15% of the patients, we identify 500 regions that were present in every single patient. So this seems to be like the core molecular alteration, or what could define CLL as a whole, regardless of the clinical behavior, regardless of the heterogeneity.